Welcome to our online course. So we're going to pick up where we left off. We're taking things in a slightly different order than they're in the syllabus for these next 3 sections, just because the order that we're doing them now I think will make more sense in the long run. So the next 3 sections we're talking about different systems of the body, organ systems that help maintain homeostasis in the body. Again, just to remind you where we are in our levels of biological organization, we're still hanging out in the systems and organs level of organization. So the last system that we talked about was the digestive system, and now we're going to tag onto that some more information about accessory glands of the digestive system in this section. So I said that we're looking at organ systems that maintain homeostasis. So first of all, we have to have a good idea or an understanding of what homeostasis is. So homeostasis is maintaining the relatively stable environment inside the body. We mentioned this early on in the semester, but now we're going to come back to that idea and look how these organ systems maintain the relatively stable internal environment and what aspects of that environment are being maintained. So first we'll talk about the digestive system, and again, like I said, we're focusing mainly on the accessory organs of the digestive system, so the liver and the pancreas. We talked about the pancreas a little bit when we were talking about obesity and diabetes, because the pancreas is what's secreting insulin, so we're going to talk about that a little bit more, and we'll talk about the role of the liver in also maintaining the relatively stable levels of blood sugar in our body. Then we'll go and we'll talk about the urinary system and the kidneys. So the kidneys are also involved in maintaining homeostasis. Kidneys mainly are maintaining the blood volume and blood osmolarity, so how many things are dissolved in the blood, and also maintaining the pH of the blood. So all of those things, the kidneys play a role in maintaining those relatively stable levels in the body. And then finally we'll talk about the respiratory system. The respiratory system we usually think of as the system that gets us the oxygen that our cells need to be able to make energy, but the respiratory system also plays an important role in maintaining the blood pH. And so we'll talk about how it does that in the third part of chapter 11 that we cover. So we're going to start out in 11.3 talking about the liver. So you can turn in your lab manuals to 11.3, and that's found on page 113 if you're using the top numbers in your lab manual. So if you need to, you can pause your video a minute and go get your lab manual so you have that with you. So first we're going to look at the structure of the liver, and then we're going to look at the function of the liver. So we've looked at the structure a little bit already of the liver when we did our fetal pig dissections. So most of you could find the liver. It's that very large organ that you see first when you open up your pig, and most of you were also able to find the gallbladder, which sits right beneath the liver. So we talked about how the pancreas and the gallbladder both play a role in digestion of biomolecules because they secrete things like the pancreas secretes enzymes into the small intestine, the gallbladder secretes bile into the small intestine, and those things help break down biomolecules and allow our bodies to absorb them. The liver also has other functions, and we're going to focus more on those other functions of the liver in this section. The structure of the liver is shown over here. The liver cells are interspersed with a lot of tubules. So the liver has a very rich blood supply. So a lot of these tubules you see through here are branches of veins and arteries. And in green here are also shown bile ducts. So a lot of the cells of the liver are producing bile, and then these bile canals lead to bile ducts. The bile ducts lead to the gallbladder. The gallbladder then stores the bile until it's secreted into the small intestine. So take just a minute, pause your video, and look at the images in your book on page 113 and identify the right and left lobes of the liver, the hepatic artery, the hepatic portal vein, and the hepatic vein. So the lobes of the liver aren't that easy to see in the figure on your book, but the right lobe is larger than the left lobe, and again, these are named right and left based on how they're sitting within the abdominal cavity. The blood supply to the liver is one of these portal systems that we've mentioned before. So I think it shows it a little better on this figure than the figure in your book. So the hepatic artery is supplying the liver with its own blood for those cells to be able to get oxygen and nutrients and get rid of their carbon dioxide waste. The liver also receives blood from the portal system that comes directly from the digestive tract. So blood first flows through the digestive tract, and then the hepatic portal vein brings that same blood to the liver before it exits the liver in the hepatic vein and goes back to the heart to be circulated again. So liver function, we're going to focus, obviously there's several functions of the liver shown here. We're going to focus on two main functions of the liver that I want you to be aware of. The first is that the liver produces urea after breaking down amino acids. So the liver plays an important role in breaking down proteins that your body is done with. They get broken down into amino acids, and those amino acids are further broken down into their component parts. Part of that are amine groups, which end up being used to create urea. So the liver is producing urea, and that's a metabolic waste product. So your body then has to get rid of urea. And when we talk in the urinary system, we'll talk about how your body gets rid of urea. But in the liver is where your body is producing urea. So on the top of page 114 in your book, it says, in the chemical formula for urea that follows, circle the portions that would have come from amino groups. So you can pause your video and do that a minute. One thing to note, if you have no idea what we're talking about with amino groups, you can go back to page 2 in your lab manual using those top numbers. And page 2 shows the structure of amino acids and points out what an amino group is in the amino acid. So if you need to remind yourself of that, go ahead, pause your video, and circle the portions that would have come from amino groups. The next liver function that I want to focus on is that the liver stores glucose as glycogen after a meal and breaks glycogen down to glucose to maintain the glucose concentration of blood between eating periods. So whenever we see that word maintain, often that is related to this idea of homeostasis. So our bodies are happiest when we have a certain level of glucose in the blood. If our bodies get above that level of glucose, then we respond by secreting insulin, our cells take in the glucose, and the blood levels go back down to normal. If that situation happens where we have a lot of glucose in our bloodstream, like after we eat a meal, a lot of the cells of the liver will take in that glucose and start building up glycogen. Remember, we talked about glycogen being one of those molecules that's lots and lots of glucose molecules all stuck together. So the liver cells will take in the glucose, build glycogen, and store it there. So then if you go through your day and it's long after a meal and you're starting to get hungry again, those glucose levels in your blood start to drop off and get kind of low. When that happens, your body again needs to maintain homeostasis. It needs to maintain glucose levels between certain parameters. And so what your liver will then do is break down that glycogen back to glucose and put that glucose into your bloodstream to bring the levels of glucose back up to normal levels. So pause your video a minute, and in that next section, number 2 on page 114, fill in glucose and glycogen on the appropriate sides of the arrows there. So we're going to do an experimental procedure. Obviously, we're not actually going to be able to do it as a class now, but I'm going to talk you through it, and then I will post the before and after pictures from doing this experiment to Moodle, so you can go to Moodle, pull up those pictures, and look at the results, and then you can fill out this section of your book. So simulated serum samples have been prepared to correspond to those blood vessels in a person who ate a short time ago, so soon after you ate a meal. We have blood from the mesenteric artery, which is the blood that is headed into the digestive tract. We have blood from the hepatic portal vein, which again is that blood supply that's coming from the digestive system, going directly to the liver. And then we have blood from the hepatic vein, which is the blood that's going to leave the liver and go back into circulation, go back to the heart. So take a second and predict which of those samples will have the most glucose and which will have the least. We're going to use the Benedict's reagent, which we've been using often this semester, to test how much glucose is in these samples. So for the results, go to Moodle and find the slides that have the results from this experiment, and you can see the before and after pictures and finish filling out that little section of your book. The next experiment we're going to look at is a similar idea, but these serum samples now have been prepared from a person who has not eaten for a long time. So now we have the blood glucose levels dropping in the body and the body's responding to that. So same 3 locations in the body, the mesenteric artery, so the blood supply leading to the digestive tract, the hepatic portal vein, the blood going from the digestive tract to the liver, and the hepatic vein, the blood supply leaving the liver. So go ahead and predict which of those samples will have the most glucose and which will have the least. And now you can go to Moodle again to look up the results for this experiment and see the before and after pictures and finish filling out that section in your book that goes from page 115 up to the top of 116. So you can finish filling out the conclusions for the blood glucose level before the next meal, and you can also finish the summary, the liver function and homeostasis there. The conclusion homeostasis that goes from pages 116 to 117 will fill out after we complete these 3 sections. So after we talk about the urinary system and the respiratory system, then you can go back and fill out that conclusion section.